Okay, so thank you very much, everybody, for coming to this session on the pharmacology of malaria and drug discovery for antimalarial drugs, success and pitfalls. I probably don't need to tell anybody in this room how serious a disease malaria is. In their last figures from 2020, WHO estimated 241 million cases globally, leading to about 600,000 deaths globally per year. That is a very major disease by anybody's reckoning. Malaria has had a very long history of pharmacological intervention. Some of the first efforts into making synthetic drugs were to make synthetic versions of quinine or to make other drugs synthetically that would be useful against malaria. And there's a very long pharmacological history and even a cultural history. You know, gin and tonic was invented to make drinking quinine a little more bearable. Efforts to eradicate malaria have been made very complicated by the rise of resistance, which keeps happening. And I think there's an appreciation now of the need to require new antimalarial medicines that target multiple life cycle stages and are often used in combination with each other, with the aim of relieving clinical symptoms, preventing transmission, and achieving radical cure, including the elimination of dormant stages in the liver, for example, in the case of Plasmodium vivax. So in this session, we have a set of expert speakers who review progress in this field and different areas of malaria pharmacology, and also tools for malaria pharmacology to make the whole process more efficient and easier. And they'll outline our current attacks against this persistent and dangerous disease. So it's my pleasure to introduce the first speaker, who is Brice Campo. He comes from the Medicines for Malaria venture. He has a long track record in drug discovery, working in Novartis and Alex Pharmaceuticals, and he's Senior Director of Drug Discovery for the Medicines for Malaria venture in Switzerland. Thank you for coming, Brice.
Thank you, Jane. I'm just going to put some water. I guess I'm going to have to... Hi, can you hear me? Yeah, okay. So hi, everyone. I'm pleased to be here. I'd like to thank the organizer and Jamie for inviting me. I got the pleasure to give a long lecture, apparently. So I'm just going to try to introduce you malaria, what are we doing at MMV, who we are, what's our pipeline achievement, and but also where we struggle and where we need to focus on the next few years to try to achieve eradication of malaria. So my name is Brice Campo. I'm part of Medicines for Malaria venture, and I'll introduce what we're doing a little bit more in the next few slides. Just a disclaimer, no need to look at that. So just to start, because I'm the first one, so I thought it'd be nice to introduce malaria to all of you. I mean, Jamie already touched a few words on that. So this is a disease that really is part of the, and touching a lot the low and middle income endemic countries. This is a disease that is taking the life of a child every 2 minutes. When I joined malaria almost 12 years ago, we were down to a minute. We moved to 2 minutes now. I mean, you could say it's small progress, but we're progressing. Unfortunately, as Jamie noted, the resistance to current treatments is developing. On top of that, COVID came along, and some of the fundings and the measures that were taken for malaria were kind of a little bit relaxed because of COVID. And the latest numbers are not very encouraging, so we need to double our effort to continue increasing this number. So this is a disease that can kill within 24 hours of the onset of the symptoms. It threatens almost half of the world's population, mainly low-income countries, Africa bearing the biggest burden. And it costs a lot of money to those countries also, 12 to 30 billion every year. As I said, resistance is spreading. We see it coming, at least for the current treatment. It's not completely desperate right now, and we are implementing measures I'm going to talk about. But this is something we need to keep in mind. Malaria has always been struggling with resistance. Every treatment that we put in the field, in the end, has gone because of resistance appearance. And this disease is both a cause and a consequence of poverty, and it really affects the vulnerable population. And by that, I mean mainly infants, children, and also pregnant women. So what is malaria? So malaria, it's a parasite that is transmitted by the bite of mosquitoes. When the mosquito bite a human, it then transfers a few parasites in the bloodstream of the human body. Those parasites are called the sporozoites. Then reach the liver, where they enter into the hepatocytes, traverse several hepatocytes until they find one where they're going to sit in and start to develop and multiply. And that's what we call the schizons. Those schizons, which are bearing hundreds of thousands of or tens of thousands of parasites, will then rupture and then reach the merocyte, released from the schizon, will then reach the bloodstream, and that's where we start what we call the asexual blood stage of the parasite life cycle. And every single merocyte will then infect one single erythrocyte, redivide, reform schizon, and then so on, so on, and so on. And that's where you start to see appearance of symptoms. After a while, some of those parasites will then differentiate into gametocytes, and those gametocytes will be taken up again by a new bite of mosquitoes, and then will develop into the mosquito into what we call the vector stage of the parasite. To ultimately form again sporozoite, which could then be transmitted to another human body by another bite of the mosquito. There's 5 different strains of malaria: falciparum, vivax, ovale, malaria, and knowlesi. Falciparum being the deadliest one. This is the main burden in Africa. Vivax being the most spread across the world, and the other ones are much less prevalent right now. One thing that we don't know is that once we get rid of falciparum and vivax, we don't know if the other ones will not then take over and then start a new cycle of malaria. An additional thing that I didn't talk about is that for P. vivax and P. ovale, both of those parasites can then enter the hepatocytes and form schizons, of course, like the others, but they can also stay dormant in the hepatocytes and reactivate weeks, months, or years after the first episode of malaria, which means that if a person has those hepatocytes in the liver and they move to a non-endemic country or an area where there is no malaria, they can restart a new episode of malaria and therefore be transmittable to the mosquitoes and then therefore to other humans afterwards. Which means that at one point, if we want to eradicate malaria, not only we need to eradicate all the forms of the parasite, but also those dormant forms in the liver. And as you can imagine, those dormant forms are very hard to reach and hard to kill. So who are we? So I work for Medicines for Malaria Venture. Basically, we were funded in 1999. Basically, on the fact that there were no pipeline for malaria drugs. So a few people from WHO then formed MMV. And our role is to discover, develop, but also facilitate the delivery of antimalarials to the people who need it. I'm not going to go too much into the detail, but basically we are what we call a private-public partnership. Our goal is to share the cost of the research with our partners, which can be both academics and pharmaceuticals company. And the goal is to then put those antimalarials on the market at a cost, basically, and making sure that those drugs reach the patient who need it. How do we work? So basically, we receive funds from donors, both governments, private donor institutions, like one of our main donors is the Bill and Melinda Gates Foundation. And then with those funding, we just evaluate research projects, both at the discovery and development stage. We fund those projects with our partner. We help them and develop those projects into drugs. And then once we have drugs, we make sure that our access department make those drugs available to the population who need it. So this is a team. We are about 120 people. We are based in Geneva. We don't have any labs. Why? Because mainly we work in collaboration, and that's why I'm showing you the map here, which is pretty much the rest of the team, which is all our collaborators. They are both from academia, as I said, industry. We also work a lot with CROs for chemistry, the MPKs. But we also work with other public-private institutions like DNDi. And we also work with research institutes like Caliber or Getty in China, for example. So we are spread across the world. Interestingly, we also work with countries where malaria is endemic. So we have a lot of collaboration in Africa. Our goal is really to develop those labs in order for Africa to be able to develop their own drugs. We had our first clinical candidate, which was developed and completely moved into human clinical trial a few years ago in Africa completely. So we hope there's going to be more in the next few years. So as I said, that was the pipeline in 1999 when MMV was created. Since then, we work a little bit with our collaborator and we filled the pipeline. So this is the current pipeline as it is right now. We have a lot of drugs now that have been registered and they are working in the field. And we have quite a lot of those drugs now which are in development. Basically, they are in clinical trial for testing. And new ones are coming into our translational and discovery portfolio, hoping that they will reach the clinical trial in the field in the next few years. One thing to notice is that 90% of our projects right now in our portfolio are geared toward treatment. So, and that's explainable because in the last 10 years, the first priority was to treat patients from malaria. Now that we're reaching this goal and we have a treatment that are working in the field, I'm not talking about resistance, I'll talk about it later. Our goal now is to try to diversify our portfolio, to try to develop drugs which will now serve to protect the population, the vulnerable population that I was talking about. And this is what I'm going to show in my talk in the next few slides. So currently our goal is to try to reduce proportion of projects which are geared toward treatment to about 50%, and the other 50% for prevention. One of the good things is that the way we develop those molecules right now, and I'll show that in the next few slides, is that until we reach the clinical phases, even when we reach the clinical phases, those molecules could still be geared towards treatment or chemoprevention, so the decision can be made later on. So the portfolio is pretty adaptable to the situation and to the future situation that we're going to face in the next few years. So the goal, those have been the goal that has been set up by WHO. So basically, the goal is to reach 90% reduction in malaria mortality and incidency by 2030. To be honest, I think this is highly optimistic, in particular with the period that we had with COVID, and try to reach eradication by 2040 and 2050. I think it's going to be not feasible, but I think we still have to live with those goals, in particular in adapting our portfolio. As you know, the drug that we're developing today will be in the field only in the next 10, 15, or 20 years. So all the drugs that we are developing today will probably serve for the last milestone of malaria elimination probably in the next 20 years. So what are we doing? As I said, we are right now focusing on preventing infections. We call it chemoprevention. And here the goal, and I'm mainly going to talk about drugs today because we are a drug, not company I would call it, but PDP. And the goal here is to really develop drugs to prevent infection in an area where there are other tools like vaccines or monoclonal antibodies, and I'll touch on that. And then our second goal is to treat patients and continue developing treatment in order to face the potential resistance that will appear in the next few years. So let's start with prevention. Here is the key population that we want to protect. As I said, we want to really focus first on children. So this is the main population that is hit by malaria. 75% of the patients that have malaria are children under 5 years old. So basically, we will look for drugs that are mainly geared for children, infants, in particular. There are some drugs currently today for chemoprevention. I'll talk about it, but they also have caveats, so we need to find new ones. Then we will also try to address the adults population, and in this particular population, we want to address the vulnerable population, such as pregnant women or the women of childbearing potential, which, as you can imagine, has some difficulties in terms of developing drugs for this particular population. And the third one are the travelers. I'm not talking about us traveling to those endemic countries. I'm mainly talking about non-immune travelers, which are included in those countries, as well as forest workers and miners, which often goes into the forest, get malaria, and come back to villages where there's no malaria, and then recontaminate people around them. So in terms of numbers, that's probably going to be around 300 million children in the next 10 years, 30 million pregnant women, and about 3 million travelers. So what's the landscape right now outside the drug environment? I just wanted to put this slide and the next one to talk about vaccine and monoclonal antibody. You've probably heard in the press a lot of stories about the vaccine. Of course, this is great news for malaria. However, this is not the solution, as it can be seen sometime in the press. It will be useful, but it will not be the only one that we're going to use to eradicate malaria. So there is 2 vaccines right now that have been either been developed and already approved, or under development and be approved in the next few years. Both of them are based on the same antigen, CSP1. They are very close from each other in terms of the technology. The key difference are the adjuvant between the two. And also on top of that, the GSK one, which is already approved, also have the hepatitis core antigen, which is part of the vaccine, while the other ones do not have this one. In terms of efficacy, they are very close, about 70% efficacy in seasonal areas and also in children. This is when you count the overall efficacy, it just dropped down to 30 to 40%. As I said, the GSK vaccine is already approved by WHO. It will roll out, it's already starting in 2022. It will continue this year and the following years. However, it's important to remember that GSK has only committed to 50 million doses in the next few years. Remember, for a children, you need at least 2 jabs and maybe a third one. So you divide that number by 3, it gives you the number of children that will be protected. And you know the number that I presented earlier is about 30 million each year that we need to protect. So as I said, it would be a very good tool, but not the only one that we need for chemoprevention. In terms of monoclonal antibodies, this is still early stage. They are still under development. Some of them are entering clinical phases this year and the following one. It seems to be a pretty good tool with a lot of advantages, in particular the long half-life, so we should be able to protect for a longer period than the vaccine. It is safe in children. The question will will it be safe in pregnant women? Those are biologics, so we hope it will be. But yes, those things have quite a few drawbacks, in particular the stability in a region where malaria is. It's very hot and humid. Those monoclonal antibodies are not stable in those conditions. And the cost of goods and treatment are pretty high also. We're talking about $5 to $8 per treatment, and you need at least 1 or 2 per year. In a low income country, this is a pretty high price. So what about drugs? So right now, there is one drug that is, or one combination of drug that is working pretty well. This is what we call a SPAC. It's a combination of sulfadoxine, pyrimethamine, plus amodiaquine. This is given once per season, every 3 months. And right now it's been shown to reduce malaria by 75% in those areas where we've been administering this combination. Last year, we protected about 50 million children, and the number continued to grow. The advantage is that it's a very cheap treatment, less than $1 per month, and total for the year is about $3.8. It's been pre-qualified. Also, the good news is that we now have pre-qualification for production of this drug directly in Africa. So Africa will be able to produce its own SPAC. And now we're trying to expand this drug to all the children, because up to now, the only children under 5 have been treated with this drug. So we're trying to move this drug now to children from 6 to 10 years of age, hoping to protect more and more children. So that, together with the vaccine, will continue to help us protect more and more children in the next few years. What about new opportunities? What are we doing next? So I will spend a few slides on the 3 pillars. So basically, we're first trying to do something on a short-term goal. What basically we're trying to do is repurpose the current treatment for chemoprevention. Then on the middle term, we're trying to recombine some of the drug that already been in the field or very close to being in the field. And then the last and the long-term goal is to try to discover and develop new chemoprophylactic drugs. I'm not going to talk about the first one, the repurpose. One of the big ones that we're doing right now is DHA piperacin, which is a treatment that is used in some areas of Africa. We're trying to do some clinical phases to see if we can use this treatment as a potential chemoprevention. So we are doing a phase 2 studies right now, so we hope to have the data next year. And I'm going to spend a bit of time on the recombine and then on the discovery side where I'm coming from. So in terms of the recombination, so one of the things we're trying to do is recombine drugs that have already been reached in the field, so pyronaridine and piperacin, sorry. Those 2 drugs are part of some of the artemisinin-based combinations that are already in the field. So we're trying to repurpose those 2 drugs for chemoprevention. So we're doing a phase 1 combo, which is pretty much almost done now, and we're waiting on the data. And then we will start a phase 2 in end of 2023 and beginning of 2024, with the goal to reach registration by 2028. So really the midterm goal. In parallel, we're also trying to develop some of the new drugs that are already in our pipeline and also already passed the phase 1 stage. We have the M5717, which is a Merck drug, and we're trying to recombine it with pyronaridine in the goal to put this molecule probably in the field after 2030. And one of the things we're doing right now is long-acting injectables. I'm not going to talk about it right now because I have a slide on that later on, and I'll show you why we are interested in that. So in discovery, our goal now is to try to develop once-monthly oral chemoprevention drugs. I come from the CNS disease area. When we had a drug that was once per day, we were very happy 15 years ago. And I moved to MMV, and one of the first things they asked me to do is at least once per week, and now we moved to once per month oral. It seems to be a little bit crazy, but it is feasible. We have some drugs right now in our pipeline that are fitting this profile. We don't have many, so the goal now is to try to see if we can enrich our pipeline with those drugs. So the goal right now is to optimize on the blood stage, like we do for treatment, because in the end, when we go in the field, we're not only going to have to protect the person which are healthy, but also asymptomatic carrier. So we're going to have to treat them at the same time that we protect them. The advantage of doing that is that once we reach to the phase 1, we can repurpose those drugs to either treatment or chemoprevention. We need very long half-life compound, more than 200 hours. We also want low resistance compound because if you have low resistance in discovery, you know that the resistance will appear as late as possible in the field. So we've optimized our strategy right now in discovery to do that. We have a working group trying to see which kind of DMPK assay we can set up or some prediction to try to identify those long-acting molecules as early as possible. This is still work in progress, but we hope to come to a kind of screening cascade that we will publish by the end of the year. We also coded all those new DMPK assay into our what we call our MMV SOLA, which is a human dose prediction tool that is available online for all our collaborators. They can enter all the DMPK and the in vivo PK parameters together with their PD parameters, and that's predicting a human dose very rapidly in order for them to see how far they are from reaching the milestone of a good dose for human. And of course, we want to invest in AI to try to help us design compound with potential long half-life. Might be utopic, but we have some compound right now in our portfolio that are long half-life, and they are looking like once monthly oral dose. We hope that we can move those molecules now in the phase 1 clinical studies for proof of concept as soon as possible. The goal now is to see how early we can identify those molecules in discovery to optimize them as quickly as possible. We're also interested in LAI, long-acting injectable. Here, the goal is not to have a monthly coverage, but at least once every 3 months, or ideally once every 6 months. We want a simple delivery, aqueous suspension, less than 3 ml. We don't want prodrug. We want the parent to be active if possible. We will target the blood stage again, like in the oral drug, with liver stage as a bonus. The advantage of LAI versus, for example, other injectables, such as monoclonal antibody vaccine, that the cost of goods are cheap compared to biologics, and we don't even need cold chain. So in terms of supply, this makes things much easier. Again, we've tried to optimize our screening cascade to try to identify those LAI. And again, we have some example of that in our pipeline right now. I'll show you in the next slides. We have 2 molecules now that are moving into phase 1 in the next couple of years, and probably a phase 2 rapidly. Those are 2 molecules that are hitting that are inhibiting the mitochondrial BC1 target on 2 different areas of the target, so they complement each other. And the advantage is that when you put them together, they synergize. So the EC50 is much more active when you put them together than when you put them alone, which helps us reduce the dose and therefore build a good formulation for LAI. So we're starting phase 1 with one of the first drug, which is atogacone this year, and the second one should start in 2024 in the aim to do a combo phase 2 studies right after that. And if that works, that would be the first LAI for antimalarial entering phase 2 in the next 2 to 3 years. So this is a landscape for malaria prevention. As I said, it's not one single molecule or biologic that will help us eradicate malaria. It's a combination of all of them, because all of them have their own benefits, and they all also have their own drawbacks. So we're going to have to combine all of them together to make sure that we move forward towards eradication as soon as possible. I just want to touch base on treatment rapidly. I don't know how I'm doing on time. Okay. So basically treatment, so this is the biggest part of our portfolio. We have treatment, they work. If you get malaria today in Scotland, you go to the hospital, you get a treatment, and within 24 hours you feel already better. Within 48 hours you feel much better, and within 3 days you'll be out of the hospital and be okay. The only problem is that those drugs in Africa or other low-income country, they're not reaching the patient. So that's what our access department is taking care of. The other thing we need to face now is that resistance is developing to those current treatment, and we need to find alternate to try to answer this issue. So in terms of treatment, what do we have? So basically all those treatments are what we call artemisinin-based combination. The most used is Coartem, which is artemether lumefantrine, which is the one on the right. This is a combination that we co-developed with Novartis. 75% of the treatment currently in all countries where malaria is endemic is Coartem. We also have amodiaquine martesonate, which is 25% of the remaining treatment, and then we also have 3 other ones that are less used right now that we're trying to repurpose for other indication. Those combinations are child-friendly. We've developed formulation, particularly for children. They are stable for 2 to 3 years in those very harsh conditions. They are approved by a stringent regulatory authority as well as WHO. And the main thing is that they are affordable. The good news is that this year, Coartem has been also approved for use in pregnant women in first trimester of pregnancy. Until now, there were no treatment available for pregnant women for this particular timing, while there was the treatment for the second and third trimester. And the good news also is that at least coartem is safe in newborns. So we can now use this treatment to try to address at least one part of the vulnerable population we were talking about earlier. In the last decade, we've administered up to 2 billion treatments, in particular, as I said, mainly coartem. This treatment is safe and is working very well. However, it's being threatened right now by resistance, in particular resistance to artemisinin. So how does artemisinin resistance manifest itself? Basically, what we observe, in particular in Southeast Asia, is that in patients with resistance, we see a delay in the speed of kill of artemisinin. Basically, instead of having a half-life of 4 hours in terms of killing parasite, we got a half-life of 6 hours. You might say, well, it's not a lot. That could be sufficient to have issues with the treatment outcome. In particular, if on top of that, you start to see resistance developing to the partner drug. So far, we've been lucky. We don't see any resistance to lumenfantrine, the partner drug of artemisinin in coartem, but we don't know when this happens. It will happen at one point, but we don't know when. So until then, we have to be prepared and find an alternate solution for that. One thing to keep in mind is that for severe malaria, so for patients where they have a very, very large load of parasites, more than 10 to the 12 parasites, they get very sick. They are very far from the hospital. We use what we call injectable artesunate. This is one single molecule, artesunate, that you inject that reduces the load of parasites very rapidly and allow the patient to reach the hospital. In cases of artesunate resistance, if you are delayed by 2, 3, or 4 hours, as you can imagine, the patient will take longer to clear the parasite. The longer you wait, the higher chance you have the patient will die. So this is why also it's important that we take care of artemisinin resistance as early as possible. So what are we doing? So this is a panel of things that we're doing. I'm going to go one by one very rapidly, just to show you we have different strategy. We have strategy where we try to repurpose some of the current combination. Then we want to transition to non-artemisinin combination. And then we have the next generation of treatment that are currently under development right now and will be on the market in the next 10 to 15 years. So in terms of current combination, basically the goal is to do better with the current tools. As I said, ACTs are highly effective. However, we need to stop or at least contain the spread of resistance. One thing that we're trying to do right now is to use what we call multiple first-line therapy. So basically using different ACTs and rotate them within the same country. It's been shown in the past that if you have resistance to a particular treatment, if you remove the treatment and kind of use another one, the parasite will become sensitive to the previous treatment again, and then you can reuse it a few years later. So the goal here is to have 2, 3 artemisinin-based combination and rotate them within the country of interest. As you can imagine, logistically, it's a challenge. However, our access department is currently working with all the governments to try to establish that. We also try to now develop tri-therapy, so we call them TACT. So right now we have one study that is called TACT, which is the combination of artemisinin and lumenfantrine, so basically coartem, together with amodiaquine. We have clinical studies ongoing right now to prove the efficacy of this triple therapy. If it works, the goal will be to develop that and put it in the field as soon as possible. The advantage that because those drugs are cheap, the overall cost will remain cheap and therefore affordable to those countries. And the third thing we're doing is trying to prevent the transmission of those resistant parasites. Recently, WHO approved the use of low-dose primaquine, which is a transmission blocking drug. So we'll try to implement this low-dose primaquine together with treatment in as many countries as possible to try to contain the spread of resistance. In terms of other approaches, there's a next generation of combination. As I said, we are worried about artemisinin resistance development. So the goal is to develop new combination where artemisinin is not present. One of the big ones that we're doing right now in collaboration with Novartis is ganaplacide lumefantrine. So we're going to reuse lumefantrine, for which there is no resistance right now found in the field, and put it with another drug called ganaplacide. We're starting the phase 3 this year. It's a huge phase 3, so it's going to take us quite a bit of our people to work on that. The goal will be to put this combination in the field as soon as possible. Something to keep in mind, this will still be a 3-dose treatment, so like the current ACTs that we're using. So, which could be an issue, because right now in the field, if you take the first dose, you already feel better. The second dose, you feel like if you have no malaria, and the third pill, most of the time people don't take it, keep it for the next time, or sell it to somebody else. That favoring the spread of resistance. So our goal at MMV is to try to develop single-dose cure where you just give one pill, and then that's it, the patient do not have to come back afterwards. So those ones will still be 3 doses, as well as the other one, the 2-drug non-artemisinin combination, like the Zydus compound with ferroquine, or the Merck compound with spironolactone. One thing to also keep in mind that those new combinations, when they will launch and be in the field, they will be more expensive than the current treatment. So something to keep in mind, which means that developing those drugs in the field will cost more. So we're going to have to find a way to continue reducing the price, or finding new funding to make sure that those drugs reach the patient. Finally, the last thing we're doing also is try to find what we call XCT, so non-artemisinin combination triple therapy. I talk a lot about artemisinin resistance. Today, it is a concern for us, but it's not as much as a concern as what's going to happen to lumefantrine in the next few years. If we lose lumefantrine, we lose pretty much all our treatment that are coming in the next 10 years in the field. So the goal today is, as I said, to replace artemisinin, or to try to protect lumefantrine from resistance development. And that's what we're doing with the triple therapy. We will try to, again, use the ganaplacide lumefantrine, which is currently starting phase 3 this year, and add the third molecule to protect both ganaplacide, which is a new molecule, as well as lumefantrine. So the goal here is to try to have a treatment that is what we call irresistible, so we cannot raise resistance. It will be very fast-acting because all those drugs are very fast-acting on the parasite, and then it will be a single dose most likely. The other good news is that all those drugs together will most likely have no impact on embryo fetal development, so we could then develop them for our pregnant women also. So switching gear now rapidly, what we're doing in discovery. In the last 10 years, most of the effort that I've shown that are now most of the molecules that are now reaching the clinic have been found using phenotypic screening. So basically, we screen huge libraries directly on the parasite, and then we see which molecule killed the parasite. We've identified a lot of hits, 30,000 hits. We've been able to deliver almost 2 clinical candidates per year, which will then reach phase 1. However, we are now kind of reaching pretty much the limit of this approach. It looks like in the last few years, we always rediscovering either the same chemical scaffold, or afterwards, when we look at the target that have been identified using this process, is always the same target that we identify. So we will need now to switch gear and try to find new ways to develop new chemical entities for killing malaria parasites. We have currently a screening cascade, or at least an array of biological assays available within our portfolio that cover the entire life cycles. And as you can see here, we have those assays are not run by MMV directly, they are run by our partners, and they are all run by academic or industrial partners. For each assays, we have what we call a center of excellence, where we can send our compounds or the compounds from different partners to be tested, same assay in the same condition, and we can cross-compare different compounds together across the life cycle. Interestingly, we also developed some what we call a clinical model, or clinical proof of concept model. Those are what we call volunteer infection model. So basically, we inject malaria to healthy patients. Of course, we monitor them, and we then test our drug of interest to see how quickly they kill the parasite. try to see if there is any recrudescent parasite, are they due to resistance or due to lack of efficacy of the drug. And with all of that together, that help us try understand from the in vitro to the in vivo to the clinic how a drug is efficacious and how can we predict the right human dose as soon as possible. Here, just to show you how good the progress have been in the last 10 years. When I started at MMV, most of the drug that we were putting and pushing into the clinic, they were around the gram scale. So basically, they were the dose. The efficacious dose was a gram for 3 days. In 2013, MMV decided that we will only develop single-dose cure molecule. When we decided that, all our partners said, well, you are crazy, we're never going to reach our milestone, it's not possible. And we say, okay, fine, let's see. We put, again, a milestone, we see if we reach it. If not, then we can rediscuss and still use the drug for 3 days. However, as you can see now, 10 years later, we are now filling the pipeline with drugs which are low single dose, so below 500 milligram single dose treatment. They are still early stage. We need to see if what we see in early preclinical development still the case in phase 1 and phase 2 studies. The preliminary data we have seems to show that it is the case, so we have a good hope. But of course, the drug can still die up to phase 2 or phase 3, so let's keep the fingers crossed. So with all of that, it means that MMV has increased the criteria, the quality criteria for all our drugs moving forward. Basically, we are now increasing stringency over time. Basically, we want to have drugs that are less than 100 milligrams single dose. We know it's feasible, so we are asking our partners to then develop all the drugs. And we will only be interested in moving a drug forward if they fill those criteria. We also increase the number of activity we want our partner to perform before we reach phase 1 clinical studies. Basically, we have now a whole range of early reprotox assessment. I'm going to talk about it in the next few slides. We also use a lot of SIMCIP files in order to do a lot of modeling of clinical efficacy. And of course, we have a whole range of high-dose PK to try to understand better the drug before we then move it into human. We have a lot of molecules going through our pipeline right now. We have many partners that I show you. So of course, the competition or in terms of funding for moving a drug from preclinical to entry into a phase 1 clinical trial is higher and higher. So I would say right now we have a rich choice thanks to our partner. And we also have insufficient resources to move every single drug forward. So we are currently in a situation where we can pick the most promising drugs, at least for treatment. We also set up a testing cascade, at least in discovery, to try to assess resistance as early as possible. I got 5 minutes. Which goes from in vitro to in vivo up to clinical trials. So we are able now to see if a drug has a high risk of generating resistance in the clinic as early as in discovery. I'm not going to go into the details. There's a nice paper from Duff et al., and you can read it. But this is coming from a lesson learned. We've developed drugs that we knew in vitro were able to generate resistance. We tested them into clinical trials, and obviously, when you go into clinical trial, they also develop the resistance already in the phase 2 studies. This is not insurmountable because we will partner those drugs with other drugs, but of course, the harder it is to raise resistance to a drug, the better. It will mean that it will last longer in the field. Finally, the last thing I wanted to talk about is that one of the things that we're really getting into now is to try to develop a drug for pregnant women and breastfeeding mothers. As I said, we now have treatment for every single stage of pregnancy for treatment. However, we still do not have any prevention for women which are in their first trimester. This is an issue because this is one of the biggest population at risk together with infants and children. So MMV is really getting into that right now and as early as possible in discovery. So we've set up a screening cascade using in vitro assays, zebrafish, and embryonic stem cell assay. I'm sure all of you know about that. The goal is to test our molecule as early as possible before candidate selection. And this will help us prioritize the molecule that has the most promise for those vulnerable population. I won't talk about target-based because Jane is going to show what we're doing in terms of target-based discovery. As I said, phenotypic screening, we're coming to pretty much an end now. So we try to find new areas to try to develop drugs, and target-based screening is one of them. We've developed and we've discovered more than 20 to 30 targets in the last 10 to 15 years. The goal now is to explore those targets, do target-based screening, and see if we can find new chemical scaffold that could be a new starting point for drug discovery. MALDA, you're going to talk about it in your next slide, so I'm not going to talk about that. And the IUPHAR malaria database, this is what Jane is going to present in her next talk, so I won't talk about that either. And with that, I just want to thank all of you and happy to answer any question.
Thank you very much, Brice. Are there any questions for Brice? Please? If you can move to the microphone, that would be brilliant because everyone can hear it.
My name is Dr. Kshir Sagar. I'm from India, Mumbai, working with the Indian Council of Medical Research. I have two questions for you. One is, you've shown a large number of drugs in pipeline, and I know MMV has been doing excellent work on that. Just question is if you have any in pipeline for the hypnozoids of the Vivax relapse stage. And the second question is, how are you contemplating to decide when you're looking at combinations? How are you looking at the partner drug when you have a new drug? Which whether you will choose an available drugs or whether you choose a new drug as a partner drug, because each would have some advantage and some disadvantage.
Yeah.
So.
Okay, so let me start with the first question. So the Vivax pipeline, yes, we have a pipeline. I didn't show it here, but we have tafenoquine that has been developed by GSK, which is a single-dose drug for relapsing malaria.
Tafenoquine, yeah.
Yeah. So we now moving with this molecule forward. It's got WHO pre-qualification, sorry. And it's now being registered in Brazil, and it's going to be used in Brazil in the next few years now. We are in the process of registering also in India, so we are discussing with some of your partners and some of your colleagues right now to try to register it in India and use it in India, and also in Southeast Asia. So with this drug, we think that we should be able to reduce the burden of Vivax quite a lot, because those are the three main areas where Vivax is. Of course, we also have South Asia, where it's still an issue there, and tafenoquine will not work very well there. So we will try to tackle that with new drug that we're also developing in our discovery pipeline. So we have several series right now in discovery that we know are killing the hypnozoites. They are very in early stage, so we hope to bring those series into preclinical development in the next couple of years, if possible. So we have a pipeline for that. It's not as big as treatment or chemoprevention, but we have quite a few things ongoing there.
If I may just interrupt, with tafenoquine, we still have the problem of the G6PD deficiencies. So
yes.
Maybe what you're looking for in the pipeline should be drugs which are effective in G6PD deficient individuals.
Yes. So for tafenoquine, it will be rolled out in Brazil together with a G6PD test. And we know that it's working. We know that if you test the patient before you give the drug, then you can give it safely to the patient. So we've shown that it's feasible, and Brazil is now implementing that completely nationwide in order to do that. the new drug that we're developing, we hope there won't be G6PD. There won't be any toxicity in the G6PD deficient patient. They are completely different scaffold. They don't have the same profile at all. So they should be safe, and they will not need a G6PD test.
Thank you.
In terms of the combination, I show a little bit of that. So it just depends on what we want to do. If we want to achieve the short-term goal, then we will try to favor drugs that have already reached the field, of course. Medium and long-term goal, we will try to mix and match. And it will depend on many factors. Of course, we want to look at pharmacokinetics, efficacy. If you combine the two drugs together, do you see synergy or not? Do you see resistance that is taking care from one to the other? This is the kind of thing that we're looking at also in terms of also potential reprotoxicity of the two drugs together or not.
Thank you. I think just in the interest of time, we have time for one more brief question and brief answer, and somebody's already standing by a microphone.
Great. Thanks for the talk. My name is Clifford Banda. I'm from Malawi, work on malaria pharmacology. One of the things that I think have come up is how the malaria community lets down pregnant women in terms of both prevention and treatment, something you have highlighted. So I wanted to ask, in terms of your artemisinin-based combination therapies, to what extent are you bringing in pregnant women to be part of the trial so that they inform the doses moving forward?
So you're right into it. So basically, the screening cascade I show you is in discovery, but we have this consortium called the MIMBA group right now at MMV, which is malaria in mother and babies, where we're trying to find assays for discovery, to try to prioritize drug for pregnant women, but also try to bring pregnant women as early as possible in clinical trial to test the drug as soon as possible and not wait for the drug to be registered, generate data for pregnant women over 5, 6, or 7 years before you decide they are good for pregnant women or women of childbearing potential. So we will bring as much as possible pregnant women in clinical studies. This is not done yet because, as you can imagine, it's very hard, but we hope to do that in the next few years.
Thank you. Thank you very much once again. So our next speaker is Beatriz Baragana. She's an apicoplexin portfolio manager at the Drug Discovery Unit in University of Dundee, and she'll be speaking for 25 minutes or so on that Drug Discovery Unit and its collaborations.
Thank you very much for the introduction. Thank you all for coming to this talk. I'm really happy to be in Glasgow. I am based in Scotland, in Dundee. So this is, for those that are not familiar with Scotland, in the east coast of the country. It's the sunny side. So if you want to see sun in Scotland, come to Dundee. And we have the drug discovery unit there. Today I'm going to present how we collaborate with others to develop new antimalarials with novel mode of actions. And Brice has given me the great introduction, but I'm going to start introducing to you what the drug discovery unit is. I'm going to briefly touch malaria because Brice has done the introduction. For those that are working at later stages of clinical development, I'm going to have a brief introduction of the early stage drug discovery pathway. Then I'm going to talk about carbamazepine. It's a compound we developed in Dundee. And then I'm going to talk about the malaria drug accelerator, going back to the very early stages of target validation and hit to lead. The drug discovery unit based in the University of Dundee is quite a unique institution because we are not really an institution formed by different groups that collaborate loosely together, but we are teams of highly integrated translational scientists across all the disciplines you need to develop drugs from hit to candidates that work together with the main aim of delivering the goal of the project and the impact. And the impact for us is new medicines for where there is an unmet medical need. When it was created in 2006, the University of Dundee and the School of Life Science created this unit, and it was really considered a high-risk strategy. There were not really many examples, if any, that in academia to do drug discovery as we do, but it's been really successful. We are sustaining investment and we are delivering impact. We want to perform high-quality innovative science, and we don't do that alone. We do it with collaborators, and we collaborate with academia in the University of Dundee and outside. We collaborate with industry. We collaborate very closely with product development partnerships like MMB and DNDi. And we also work closely with funding bodies like the Bill and Melinda Gates Foundation and Wellcome. Our aim is to translate the best research into commercial opportunities, and we have been successful in doing that, and I'll show you later some of the numbers around the DDU. But we're really very passionate about developing high-quality leads and drug candidates for a medical need. And this is our mission, and to do that, obviously, we need to provide a supportive and excited environment for our staff, and we are keen on publishing and also disseminating knowledge so people don't repeat things that don't work and learn from the things that work. So the drug discovery unit in Dundee in numbers is here. So we have developed 5 compounds that we've taken into clinical development. Our end goal is clinical selection, and after that we partner. We don't run clinical trials. We have 130 members of staff that are not postdocs, are members of staff that work permanently with us and that are integrated in teams. They're very experienced, the translational scientists. We have delivered 9 assets to pharmaceutical companies. Of those, 5 have gone to new companies, helped them to start. And since 2006, we have gained £100 million in funding. The drug discovery unit is divided or organized, better than divided, in 5 portfolios. 4 of them are anti-infective. We have a really strong commitment to anti-infective diseases, particularly those that affect low middle-income countries, particularly neglected tropical diseases. And then we have a portfolio that is based on working in novel targets or novel pathways. This is disease agnostic, but we currently focus on neurodegeneration in that portfolio. I lead the portfolio that is called the Picoplexan portfolio. It started being the malaria portfolio and is still very heavily involved in malaria projects. Okay, malaria has been introduced now 2 times. This is going to be the 3rd time you hear about the impact of malaria. It's true that malaria is preventable and curable, and it's really true all the numbers that have been reported by WHO in 22. We still have a high number of cases. We still have a high number of deaths, mainly in children in Africa under 5. And although there's been control and improvement since 2000, and I always remember what Brice said, when I started working in malaria in 2010, we used to say that a child died every minute, and now we say every 2. But still, the reduction, what is worrying, is a stall. And since 2015, the curve was going down, and now is flat or going up. So we need to do better. And as Brice mentioned, we worry about potential of artemisinin resistance that we see in Africa and can see in Asia and is spreading to Africa. And we're still not really sure of the consequences that this is going to have. We want to control malaria, but that's not enough. Really, we're looking at eradicating malaria, and we recognize, WHO, all the partners, MMB recognize that for that we need to step up innovation. None of the tools we have now alone is going to be sufficient to eradicate malaria. And I think we also recognize that we need a combination of prevention and new tools to be able to do this. And in the area of new medicines, we're really looking, as Brice said, for single-dose cures that are also, they don't have resistance in the field. We're looking at compounds that can block transmission, so we stop the spread of the disease, compounds that are capable of chemoprotection, and Brice describe really well their strategy for MMB and they prevent relapse. And the center of this is our target population. We're targeting children. We need pediatric formulations. We are also, these medicines are going to be also very important for pregnant women and women of non-pregnancy status, and we need to be safe in pregnancy with these new molecules. So how do we connect these aspirations that we have into what we do in the early stage in early stage drug discovery? So we need target product profiles, and I think Brice touched on that. They're really excellent at developing target product profiles for malaria, and they do that with experts and clinicians. And then all us that we do in drug development for malaria have this framework that is really needed. We need to start with the end in mind, and we need, from that, we can distill compound progression criteria. And this is very important because we have all these stages to get from identifying a hit to delivering a candidate. And to know when we are making progress and when we can go from one stage to the other, we need this compound progression criteria. I mean, just for those that are not in early stage drug discovery, I'm going to go very briefly through these stages. So we start by identifying a hit. This hit is identified through developing an assay that could be a biochemical or a phenotypic assay. And then the hits usually are really a good starting point, but not even good to put in a mouse model of efficacy. And we need to start the hit to lead phase, where we're looking for compounds that we can progress into a mouse model to demonstrate that this phenotypic hit or this new target has the effect in a mouse model. This is the hit to lead. If we then move into lead optimization, there we're looking for much better molecules where we can have predictions. We use MMB SOLA that Brice just mentioned, a predictive model that tells us what the human PK is going to be, and also the dose. And we're looking for very low doses. As you see now, we're targeting less than 500 mg, but ideally less than 100 mg. And obviously, these compounds had to have low toxicity. And then moving into the clinical candidate selection, we obviously evaluating much more stringent evaluations of safety and in vivo. And in the DDU, at this point, we partner with MMB, with pharmaceutical companies, and they take compounds in the clinical trials. And just the final kind of brief introduction of what we do in hit to lead and lead optimization is a difficult task. It's not really trivial. We're not optimizing one or two parameters. This is a multi-parameter optimization where we're looking at potency, selectivity against the human target, physicochemical properties, pharmacokinetic properties, and we are now very early start with in vitro safety, cardiovascular safety, drug-drug interactions, mutagenicity, and others. So we're not looking for the best molecule in any of these quadrants. We're looking for balance of a molecule that has a balance of all these properties. And we monitor this with the compound progression criteria that just mentioned comes from the TTP that MMB published. We started working in malaria in the DDU in 2009, and we started really very simply. We started with developing an assay that looked at how to kill parasites in vitro in the asexual blood stage that Brice just mentioned is the stage that causes the symptoms of malaria, and obviously we need to clear parasites in the asexual blood stage to cure malaria. We screen a very small library of only 5,000 compounds, and we identify 120 hits that we run in those response curves with 10 different concentrations. And then looking at the chemical structures, that's what I do because I'm a chemist, we classify compounds by the chemical structure into what we call series or clusters. And from those, once MMB at that time checked what was novel and told us 3 of these compounds, chemical series were novel, and they gave us funding for the screen, but also to take these compounds further into the hit to lead. And I'm going to talk about MMB04 series, that is a series that progressed further of these 3. So we had a hit, and this hit was fairly potent. As you see there, the first row is potency 100 nanomolar, that's okay for a hit. But it had not really very good solubility or microsomal stability. We're looking at an oral drug, and therefore we need to be soluble to be absorbed, and the compounds need to be stable once they're being absorbed to have enough concentration to have an effect. And at this point, we're not really looking at improving potency, but we spend time looking at improving the properties. And to look at see if we were making, to check that we were making progress, we're using in MedChem an efficiency metrics that one of them called lipophilic ligand efficiency, that is an assessment of the compound quality. And we want to see that number, this LLE, going up. And as it goes up, it's telling us the compound is not only more potent, but it also had the properties good properties to be an oral drug. When we make changes in the most lipophilic areas and go to the third compound in that table, we were happy because we have a soluble compound and we have a metabolically stable compound, but MMB, our partner, started having doubts about the project because we had been working for a little while and we were not making improvements in potency. And that was a critical point because we had 6 months to demonstrate that we can improve the potency of these molecules. And the day that I prepared the early lead was really a very happy day because we were making a few changes in that area there, and we identified that we can make changes that improve the potency, and now we have a 70 nanomolar compound that also had good properties to go on in vivo in a mouse model. And this molecule demonstrated efficacy in a mouse model, so it gave us more time to work in the project. However, this is not the perfect molecule because the bioavailability when dosed orally was not good enough, and they require a high dose for efficacy. And we demonstrated that the low bioavailability was caused by a low permeability. This molecule was soluble but not very permeable and couldn't go through membranes. So we work more in this series and we identify 498. This is the first name the molecule had. It's our preclinical candidate. It's very potent and had really good balance of all the other properties, leading to good bioavailability across species, mouse, dog, mouse, rat, and dog, and good efficacy, including a single dose cure in a mouse model of malaria. To achieve this, we prepared 300 molecules. And this obviously is a team effort of chemists, biologists, DMPK scientists, and pharmacologists. At this point, we hit the preclinical candidate selection phase, and Brice described how they put together lots of assays to test the activity of the molecules across the life cycle. And this molecule became really interesting for everybody because it had activity 1 nanomolar in the sexual blood stage, both in falciparum and P. vivax, but it was also active in transmission blocking stages, so therefore had potential to prevent transmission from human to human through a mosquito bite. This is, as we mentioned before, very important for eradication. And it was similarly potent in liver stages, and this is one of the things that probably now is not so important in chemoprotection. At that time, it was one of the requirements to set a molecule for chemoprotection, and it's very active in liver sisons. So this was interesting because at that point, there were not very many molecules that had this similar activity across all the life stages. And it also was interesting because this molecule didn't show cross-resistance with any of the non-molecules at that moment. It had also other exciting properties, so it was predicted at this point that it could be a single-dose cure agent, and at that point we predicted that 800 mg could be curative in an adult. It had a good therapeutic ratio, and also, very importantly, it's a molecule that you can prepare in not very many steps from cheap starting material, so the cost of goods is very low, and it doesn't require expensive formulation because it's a very soluble molecule. So it ticked all the boxes, and with this life stage activity and also not being cross-resistant, we were very interested in knowing what was the molecular target of this molecule. We had get to this stage with a pure phenotypic approach, and we didn't know how this molecule killed the parasite. So we did this study of mode of action of 498 in collaboration with Julian Reiner, that at that time was in Sanger, David Fiddock in the University of Columbia, and Marcus Lee, that at that time was a postdoc in David Fiddock lab, and is now with us in the University of Dundee as a professor. So what they did was resistance selections and then whole genome sequencing to demonstrate that 498 act by inhibiting elongation factor 2. And what does elongation factor 2 do? Elongation factor 2 is responsible for the translocation of the ribosome across the messenger RNA. So when we block this effect, what we're blocking is the protein synthesis in cells. And this is selective, so it's not. It's only inhibits elongation factor 2 in Plasmodium, not in humans, but also explains why it had activity across all the life stages of the parasite, because in all of them, protein synthesis is essential. At this point, we gave back the molecule to MMV, and MMV found a partner, a pharmaceutical partner, and Merck took the compound into the first-in-human clinical trials. They ran a randomized, placebo-controlled, double-blinded, single ascending dose study, and there it showed that the half-life we had predicted that was going to be long in humans is effectively long, and also that is well tolerated. With this long half-life and the time that was above the minimal parasitic concentration, it gave really a good suggestion that this molecule was going to be a single dose, suitable for single dose cure. And this was demonstrated in the phase 1b study. It's a human volunteer infection. And in this study, human volunteers are infected with malaria, treated with single dose of 498, or the name that Merck gave him, M5717. And the 800 mg dose, a single dose of 800 mg, cured malaria in these volunteers, and there was no recrudescence observed. And this is very exciting because, as Brice showed, the current treatment is a three-dose treatment. And the effect of reducing 3 days to 1 day has a much better possibility that the patients are going to be compliant and potentially had an effect in controlling resistance. So the 498, or M5717, is now called cabamyquin, and is gone to a couple of other studies. So Merck sponsored a phase 1b study to assess chemoprophylactic activity with a single dose of cabamyquin. It's a sporozoite human challenge. This is completed, and the results are positive, but I don't think they're published yet. And then is now currently in Uganda undergoing a phase 1a combination study. This is for acute and complicated malaria, and it's a combination with pyronaridine. Obviously, we do a combination to prevent resistance, emergence, and the product vision is treatment with a single dose to manage resistance. And it also has potential for chemoprevention, as Brice mentioned. One advantage that cabamyquin has is that it's proven to be safe in preclinical models of reproductive and embryofetal safety, and therefore it has potential to be used in pregnant women and in mass drug administration. So for this part of the talk, I want to thank the team in Dundee. We were a very small team at that time, and we couldn't have done this alone. As you could see, when we published this, we published with 70 authors in the paper. That means that we had lots of collaborators, both in academia and industry. To achieve this, you need really a very big team. And I'd like to thank MMB because they gave us all the funding from start to clinical selection, and obviously Merck for realizing the potential of this compound in the clinical trials. So I'm going to switch gears now, and I'm going to go back to the very start of drug discovery. Brice mentioned that malaria, we have done very small effort, but the community has done a very big effort in phenotypic screens, and millions of molecules had been screened. And now we're thinking about using more a target-based approach, but using the information that we have from these phenotypic screens. If you want to do a target-based approach for drug discovery, whatever the disease indication, you need to select targets that are robustly genetically and chemically validated. If not, it's going to be a failure. You're going to be waste a lot of money to fail because your target is not validated. And obviously, in malaria, we need to be very careful we don't do that because the funding is not really as good as in other areas. So we are part of the Malaria Drug Accelerator, or MALDA. This is funded by the Bill and Melinda Gates Foundation, and we are 18 institutions, including MMB, including pharmaceutical companies, and also institutes and academic groups. And what we want to do here, and we're aiming to do, is to validate novel molecular targets for malaria treatments. Very often we start with phenotypic hits, and I'll show you an example later. And once we identify these molecular targets, we select the best ones that we think have promise to fit the target product profile set by MMB, and we go and identify and optimize inhibitors for this target. And this is where my team collaborates strongly with MALDA. These projects are all collaborative and very open, and typically a MALDA project has 5 or 6 sites working together. So I'm going to present very briefly, just in 2 slides, one of the projects I've been working with MALDA, and it's the chemical validation of acetyl-CoA synthetase. And this started with a phenotypic hit, this molecule, MMB721. This was part of the Tres Cantos antimalarial set, that is a big set of phenotypic actives in malaria. This is all blood stage malaria. But it also had activity against other stages, so it caught the attention of the MALDA parasitologists, and they decided that this was a good molecule to start looking for a mode of action. It also had a fast rate of kill, what is a positive, because it can kill the parasites as fast as chloroquine. So what they did was to go through resistance selections to look at identifying parasites that were less susceptible to this molecule, and then ran whole genome sequencing to see what the mode of action was. And they identify single nucleotide polymorphisms in the gene encoding for acetyl-CoA synthetase. This is a novel target for malaria, and it hasn't ever been expressed recombinantly. My team did the recombinant expression of full-length acetyl-CoA synthetase, and we developed assays to measure inhibition. And we demonstrated that 721 inhibit the wild type acetyl-CoA synthetase. We also express 2 proteins carrying the single point mutations we've seen in the resistant clones, and demonstrated that at the protein level, it was also, this 721 was also less active against these proteins. And we expressed the human ortholog, and we were very pleased to see that the inhibition was selective for the recombinant Plasmodium versus the human. And we published this with our collaborators in cell chemical biology last year. What we've done since then is taking this molecule, 721, that was a compound with a moderate potency and not really very good solubility or metabolic stability. As you've seen before, this is typical of a hit, and take it into the first phase of optimization, the hit to lead phase, improve the potency, and obtain new molecules like 405, the early lead, that is metabolically stable and soluble. And that allows us to then demonstrate that this series has potential for the efficacy in a proof of concept in a mouse model where we dose it orally at different doses. We've seen in some of the doses the parasitemia goes below the limit of quantification. We did this again with around 300 compounds, and we also in the process of obtain crystal structures of these molecules bound to acetyl-CoA synthetase. So we now have a structurally enabled project. And the project is now ready to enter lead optimization, and we have been successful of obtaining Medical Research Council UKRI funding to start this month the lead optimization, and we're aiming obviously to increase the half-life. We need to go as quick as possible to this month, but I don't know if a month is going to be achievable. We'll try. We need to increase potency and safety, and prove safety, because so far we don't have flags, but there are many things we need to test. And we want to, in 30 months, have a compound that is suitable to be entered into preclinical selection. I'd like to thank the team in Dundee, really brilliant scientists, very committed, and then obviously also to all our collaborators, and Bill and Melinda Gates Foundation for funding, and our MALDA partners that worked with us in the validation of this target. So with this, I have concluded. I thank you for your attention, and I'm ready for any questions you may have.
Are there any questions for Beatrice? As people collect their thoughts, can I ask a slightly broad question?
Yeah.
But you talked about moving from your first hits to improving the molecule with your changing rings and changing halogens and so on, and having to test lots of molecules. We hear lots of either exciting news or hype, and I don't know which, about the role of artificial intelligence in guiding this process to make it faster. When I read this stuff, should I be excited, or should I just think this is all hype and it's all going to be really difficult the way it has been for you?
So we are we are also applying AI, starting to apply AI in the DDU, but obviously a much lower level than companies came from that lead, like Exentia. I think you should be excited about it, maybe a bit cautious, but I think it has a lot of potential. Very important for that is to have high quality data. So, and also data that is consistent. And this is part of what is going to drive, if we want to have good, if they want to be effective, is high quality data that is consistent, that can fit the models, and then the results are going to be high quality results. If the data is not good and well curated, the AI model is never going to work. But I think, yeah, it has a lot of potential.
Thank you. That's reassuring. Are there any other questions for Beatriz? In that case, thank you very, very much for telling us about that. And it's my pleasure to welcome Aida Kawuma, who is a pharmacometrician at an infectious diseases institute working in Uganda.
So thank you very much. I'm excited to be and share my work. And as a potential end user of the medicines that you're developing, it's really exciting to share my work. So one of the questions that as a visibly pregnant woman and one who has breastfed before, one of the questions I always ask my physician when I go to an appointment is the medication you're prescribing for me safe? Is it safe for me? Is it safe for my unborn child? Is it safe for my infant who's breastfeeding? And so to just contribute to that body of knowledge, my co-authors and I undertook a systematic review to talk about maternal-to-infant transfer of drugs through breast milk during the treatment of malaria, tuberculosis, and neglected tropical diseases. So malaria, as you've heard, and TB, together neglected tropical diseases, are prevalent in regions where breastfeeding really is the only acceptable, feasible, accessible, safe, and sustainable option. And so that means that we will have pregnant and lactating mothers who are breastfeeding and on these medications. Because in 2021 alone, the WHO reported more than 300 million incident cases of malaria, TB, and neglected tropical diseases combined. Unfortunately, we still have limited knowledge on the extent and consequences of infant exposure to drugs through breastfeeding. Clifford asked Brice a question about the fact that pregnant and lactating mothers are so often left out of early clinical trials. And as a result, we hardly ever have any information until the drug is already being used. So what was our objective? We set out to summarize the body of knowledge on mother-to-infant transfer of drugs used to treat malaria, TB, or neglected tropical diseases through breastfeeding. We also wanted to understand the different mathematical approaches that are used to characterize this transfer. How is it characterized? How is it described? Because that leads to evidence on infant exposure. Lastly, we also wanted to provide recommendations of analysis on lactation pharmacokinetic data to improve the characterization and exposure of these drugs to a breastfed infant. So why this is vitally important is because, as they've said, if we don't know how much an infant is getting through that breast milk, that means they may either be predisposed to very suboptimal concentrations, low concentrations, which might select for resistance, or they might be subject to toxic concentrations. We really don't know. So what was the process? The World Health Organization has a list of treatment guidelines for malaria, tuberculosis, and tropical diseases. So we went to WHO, the treatment guidelines, and generated a list of drugs that are used. Once we had that list of drugs, we had 2 independent reviewers undertake an extensive search in PubMed, Scopus, or LactMed. There was also a 3rd reviewer who acted as a tiebreak in the event that the 2 reviewers didn't agree. So we came up with a total of 660 records, publications that talked about lactation pharmacokinetics for either of the drugs that we had listed. After accounting for duplicates, we narrowed that down to only 327 articles and publications. And so these included full-length articles, this included conference abstracts, and That came down to 53 articles, but eventually we only included 18 studies in our qualitative synthesis. So what were some of the reasons for moving from the 53 we had down to 18? So some of the articles we saw were review articles, and since we're doing a systematic review, they weren't included. Other articles only had abstracts available, so we did not have access to the original articles. And some were just case report studies with maybe 1 or 2 participants. What did we find? Like I said, we started with the list that had been generated from WHO treatment guidelines for either malaria, tuberculosis, or neglected tropical diseases. That list consisted of 69 drugs, so both for drug-susceptible TB and drug-resistant TB, complicated and uncomplicated malaria, and a whole slew of neglected tropical diseases. So those were 69 drugs. Interestingly, of the 69 drugs, only 40 drugs had any information of concentrations in breast milk. And surprisingly, of the 40 drugs that had information on lactation pharmacokinetics, less than half, only 15 drugs, had information that had been derived from lactation pharmacokinetic studies. And that's a very big disparity because although 40 of the drugs had some information available, most of the information came from activities like bioassay development or studies that had been designed for a completely different purpose. So you ended up with single time points, really studies that were not informative at all, no clinical data, so you couldn't include them among the 15. And so by and large, it came down to only 15 drugs with lactation information, lactation pharmacokinetics information. And this was available from 18 lactation studies. And so what we did, we then looked at these 18 lactation studies and scored them, evaluated them, vetted them using what we call a clean PK checklist. And I'm going to talk about that later. But basically, this clean PK checklist seeks to give guidance on how clinical pharmacokinetic studies should be reported so that we have some sort of conformity. But the key message here is that only a fifth of WHO recommended drugs have lactation pharmacokinetic data. And so that's a big challenge for teams like MMV and that are trying to develop. How can we bridge this gap so that by the time we have drugs that I can use and I can feel safe using, we have enough information on them. So obviously, I'd like to sort of begin to wrap this up and say that we found that there's a scarcity of lactation pharmacokinetic studies, with only 58% having information regarding breast milk concentrations. And 66 of these studies used non-compartment analysis methods and derived the milk to plasma ratio to estimate infant exposure. So usually the milk to plasma ratio is derived by, you get exposure in breast milk vis-a-vis exposure in plasma. So that's what we are calling area under the concentration time curve. But also, and I'm glad that Brice mentioned it, we are beginning to see model-based analysis, which is compartmental methods, are increasingly being used to predict infant exposure. So this includes what we call pharmacokinetic, pharmacodynamic modeling, physiologically based modeling, and I'm glad that in both presentations earlier, they showed components of that. So that might help speed up the pipeline. I also want to draw your attention to this clean PK checklist, against which we scored the studies. So it's a 24-item checklist that is used, that gives guidelines on how to report lactation on clinical pharmacokinetic studies. So by and large, we found that it was inadequate for reporting lactation pharmacology, especially in low and middle income settings. This guideline was developed using what we call a Delphi methodology and was developed by scientists really in high income settings where in critical care conditions like high dependency units. And so some of the criteria that were on this checklist are irrelevant for many of the studies that we looked to. For example, this checklist requires you to describe in depth the different approaches you used for extracorporeal drug removal. And that would be like in a high dependency unit, which many of these studies didn't report. So by and large, this clean PK checklist was somewhat irrelevant and inadequate to assess the 18 studies that we looked at. So I want to conclude by saying there's need to build consensus and establish best practice guidelines for conducting and reporting lactation pharmacokinetic studies. Again, I'm delighted that MMV have said they are wanting to incorporate pregnant women and mothers who are lactating much earlier. But there needs to be a consensus on how to do it, what's the best time to do it, and how do you report that. And then we've also seen, as they've shown, that compartment analysis, which is modeling basically, is a useful tool in understanding plasma to breast milk transfer of drugs, and we should increasingly use it to help speed up the pipelines. So with that, I'd like to acknowledge my co-authors, Dr. Francis Ojara, who unfortunately couldn't make it because of visa restrictions, Professor Katrina White, who's in the room, the EDCPT Virtual Consortium for my travel support, and the committee for letting us share our work. Thank you very much.
Thank you.
Sorry, I would like to say that currently we have a protocol in Uganda, the Infectious Diseases Institute, where we're actually recruiting lactating mothers, and we are actually looking at drugs for malaria. So if you're interested, please see me after for a flyer. And interestingly, the study is called Milk. So,
Thank you very much for a very interesting talk. Are there any questions for Aida? Well, can I start with a question? And I'm sorry if I'm supposed to know the answer to this, but how different are humans in lactation to animals like cows or whatever? I'm just wondering how much of the basic PK you could get from animal models, or are we very different?
that's an interesting question. So I think one of the things is the debate around foremilk and hind milk, for example, and the composition thereof of it. And when you're taking the studies, so one of the challenges is when you're taking lactation, when you do lactation PK, there isn't any agreement on how best to quantify how much the infant takes in. So there are estimates about 150 mils per kilogram per day, but there isn't really, all the methods we are using are not optimal. There are different methods such as empty the breasts, so there are different methods. And so if we are not doing it, if we're not able to do it well, even within the humans, it might be a bit more challenging to translate from an animal model in lactation to a human model. But Brice did mention something that we call physiologically based modeling, pharmacokinetic modeling. And that is, I think, would be closer to mimicking humans, because now there's a lot of information about transfer rates, about the contents of the breast milk, about the permeability of the drug, and I mean the permeability, the solubility of the drug. So the answer to your question is difficult.
Okay,
Yeah.
Thank you. Thank you. Are there any other questions? Okay, well, thank you very much for a very interesting presentation and for coming a long way. Coming less of a long way, although ScotRail can make it feel that way, is Jane Armstrong, who is a senior curator of the IUPHAR MMV Guide to Malaria Pharmacology database.
Thank you, Jamie, and I'd also like to thank the organising committee for inviting me to present to you today the IUPHAR MMV Guide to Malaria Pharmacology, which is an expert-curated database of Plasmodium molecular targets and the compounds that act upon them. And just to say, that's the URL for the database underneath my name. So in my talk today, I'm going to start by giving you a little bit of background to the parent database, which is the Guide to Pharmacology, before moving on to the Guide to Malaria Pharmacology itself. And then I'll finish my talk by taking you through using the guide, hopefully demonstrating how to navigate and view our malaria-relevant content, and also highlighting a few new features that we developed for the guide. So the parent, so the Guide to Pharmacology, it's a free-to-use open resource, most important to note that, and it was formed from the merger of the IUPHAR database and the BPS Guide to Receptors and Channels. And this formed a database of human drug targets and the prescription medicines and experimental drugs that act on them. For curation, all the data are manually curated and they're selected from the published literature. And also to note that the data are reviewed by the subcommittees of the IUPHAR Nomenclature Committee, which is an international network of over 1,000 scientists, these subcommittees. Moving on to the content of the Guide to Pharmacology, we have over 3,000 human targets. On the left, you'll see a breakdown of the categories of the targets there. We have over 1,900 targets with curated interactions, and over 700 that are targets of approved drugs. And also just to highlight here that we have a very large number of references in the database. For the ligands, we have over 11,000. Actually, looking at those numbers there, we're approaching 12,000. So I think at the next database release, we'll go over the 12,000. And of those classes, the majority of these are small synthetic or semi-synthetic ligands. We have over 1,800 approved drugs, over 8,000 with curated interactions, and over 3,000 with clinical use summaries. Database access statistics, I'm not an expert on this, but if you look at the number of sessions, the number of users, and the number of page views over, and look at that for the previous 12 months versus the 12 months before that, we've had a steady increase. And then if we look at the number of, on the right-hand side, the top blue line is the number of sessions in the previous 12 months, with the orange is the 12 months before that. So there's been a sustained increase over that period. The Guide to Pharmacology's homepage is a user-friendly way to access all this data. The user can browse either through the quick links on the left-hand side of the page or using the menu bar. We have a search on the home page and an advanced search, and also on every page in the database there is a quick search up in the top right-hand corner. On our homepage, we also have links to news and the popular hot topics, and also we highlight recent publications or conferences coming up. So this homepage has been designed for the needs of pharmacologists, and when we wanted to add, we're adding antimalarials, we realised that we needed to organise the information in a slightly different way, more tailored for the needs of that research community. So with that in mind, and with funding from Medicines for Malaria Venture, we developed the Guide to Malaria Pharmacology, with the aims of extending the content in the Guide to Pharmacology with curated antimalarial compounds and the Plasmodium molecular targets, and to provide a new portal that was optimized for malaria researchers. The portal itself was given a complete identity, and you can search across the database. There's a search in the top right-hand corner, and that upweights antimalarial data. And then we designed the homepage to have these panels that you can browse the data with. Two of those, we've kept the targets and ligands panels, but then we've introduced two new ways of browsing the data, which are the parasite life cycle stage and the target species. And just to mention, these categories can be accessed from the top menu panel as well. Sorry, it's top menu bar. This is just to illustrate that behind these two guides, there is a single database. And for curation, all the data, again, has been manually curated from the literature with the guidance from our committee of scientific advisors. Our curated ligands include antimalarial leads, drugs in preclinical and clinical development, approved medicines, and we also include compounds that we've picked up from patents as well. At a most recent database release, we have just over 135 compounds with antimalarial activity and 40 Plasmodium targets. So I thought I would finish with a quick guide of how to navigate and find and use the database. And what I'll do is I'll take you through each of these 4 ways of browsing the data. If we start with the targets, if you click on the panel, there's antimalarial targets, and that takes you to the antimalarial targets family page, which has a brief description, and then the targets are categorized into subfamilies. And if you go into each of these categories, there's links to the individual targets. And if you click on one of those, you come to our detailed target page. This is modeled on the Guide to Pharmacology's detailed target page. It shows all the curated information that our database holds about the target. At the top of the page, you can see gene and protein information. We have links to other databases, such as PlasmoDB and UniProt. And then there's also, if there's 3D structure information, that's included. And then one of the new features is a comments section that the curators can add information relevant to the target in that box. Going back to the home page and to the ligand box, if you click on the all antimalarial drugs, you'll get the ligand list page, which lists all the ligands in the database that have been tagged as antimalarial. And if we click on one of those, we get to the ligand summary page. So again, this is modeled on the Guide to Pharmacology page, and it shows all the curated information for that compound that the database has. If we look at this page in a little bit more detail, the top section on the left has the key information. There's synonyms there. And then, in the previous slide, you may have noticed little icons. Going from left to right here, this is telling you the drug is an approved drug, that it has a structure in PDB, and then the third icon is to show you that it has antimalarial activity. There's also curator comments in that section. And then on the right is the 2D structure. And we have drop-down menus below that give you information, structural information such as inch key, and the smiles can be found there. And then underneath that, we have a series of tabs. The summary tab is open by default when you come to the page, and within that you'll find links to other databases such as PubChem and Chembl. And then there's a series of additional tabs. I'd just like to demonstrate the Malaria tab, which is one of the new features that we've introduced for this guide. And this allows the curators to add comments, so just a very brief one here. They can be quite extensive. And we also include the potential target and mechanism of action here. Quite a lot of the antimalarial drugs will have an unknown mechanism of action, and that'll be noted there as well. Just to go on to one of these, browsing, one of these new ways of browsing, the parasite life cycle stage. There's a list of the life cycle that we've divided it into 5 top-level stages so that we can annotate the data in the database and organize it and bring it back from searches. At the bottom, there is a link to what we call a homepage that has background information about the Plasmodium life cycle and links out to more resources. And then below that is these 5 stages, which are also available from that main panel. And if we click on one of these, so in this case, the Plasmodium sexual blood stage, this brings back the page for that stage, which includes the total number of target and ligand associations for that stage that are in the database, a brief description of the life cycle stage, and then a table providing a complete list of all the interactions that we have in the database that's been filtered for this stage. And from that, you could click on the target, or you could click on ligand. In this case, click on ligand, and we get back to the ligand page that we looked at previously. And while we're on this page, I just wanted to demonstrate one of the other tabs, which is the biological activity. If we click on that, it opens up the tab, and here at the top, you'll see we have quantitative information about the human target, and below that on the parasite itself. And if we click on the Plasmodium target link, that takes us back to the detailed target page that we looked at earlier. And on that page, you can also find all the other ligands that we have interaction data for that particular target. And each of the rows can be opened up and to give more details, so about the assay and about the Plasmodium life cycle stage that it's annotated for. And then the final box is the species of the parasite. Again, we have a page with a little bit more description that's available, or you can click on a species itself, and this page is organized in a very similar way to the life cycle stage, in that you have the total number of targets and ligands that are associated with this stage, and then a section for a brief description, and then a table that provides a complete list of all the interactions we have in the database. And then you can click through again to the target or the ligand and find out more information about any of them. I'll finish there, but the best way to find out about a database is to go and give it a try. So just a reminder that this is the URL, if you want to go and try it. And then to find out a little bit more about the Guide to Malaria Pharmacology, we have a poster presentation tomorrow, which is for the guide to pharmacology, but there's a section for the malaria. Simon Harding will be with that tomorrow lunchtime. We publish in nucleic acids research every couple of years, the database issue, and both the 2022 and the 2020 update has quite a bit of information about the Guide to Malaria Pharmacology. We have a help page available from the home page. We have blog posts. You can follow us on social media, and you can email us at any time. I'd also finally like to just highlight that we've recently published an IUPHAR invited review. This was with MMV and the MALDA consortium, and it's focusing on recent advances in target-based drug discovery, but there is a section included on the Guide to Malaria Pharmacology. And then just to finish to thank everybody involved in this project. The database team are based in Jamie Davis's lab at the University of Edinburgh. I also wanted to thank all our scientific advisors from NC, IUPHAR and MMV, and also the MALDA consortium, the authors from the review that I've just on the previous slide, who helped with target descriptions that are included in the database. And this is Edinburgh looking rather beautiful in the snow, how it normally is. And I also just wanted to thank all the funders, and also thank you for your attention today. Thank you.
Thank you, Jane. Just as we take questions, may I suggest you put the slide with the QR code on, because a few people, I think, were about to photograph that when it disappeared. Thank you. Are there any questions for Jane? Well, can I ask you a very general question, which is, if people have got any ideas of something that should be in, or any suggestions about how to improve the database, how do they contribute?
Well, that email that I showed you, that is at the bottom of our pages, is it? Yes, it's the bottom of, I think, every page of the database. So, and yes, we'll respond to that, and we would love feedback and information. That would be fantastic.
Thank you. Are there any other questions for Jane? Okay, in that case, thank you very much again. And we come on to our last talk of this session, and that's going to be from Koketso Mogwera for targeting the Plasmodium kinome for malaria drug discovery.
Okay. Good evening, everyone, and thank you very much for the invite. It was Kelly that was supposed to be here, but yeah, he saw fit that it should be me here. Yeah. And as already indicated on the screen, the title of my presentation is Targeting the Plasmodium kinome for Malaria Drug Discovery. So I will go into a brief introduction of H3D, the organization, and then a brief history of MMV048, and then a short case study about mining the kinome before I conclude. So who are we? We are an accredited research center within the University of Cape Town, founded in 2010. And our vision is to be a leading organization for integrated drug discovery and development. So our mission is to discover and develop innovative life-saving medicines for infectious diseases, build Africa-specific models to improve patient outcomes for African populations, to develop different platform technologies and train African scientists in drug discovery-related sciences. So we have different technology platforms, some housed at the Faculty of Health Sciences and others in the Faculty of Science. So within the Faculty of Science, we have the medicinal chemistry, computer-aided drug discovery, AI and machine learning platforms. And then within the Faculty of Health Sciences, we have DMPK and biology. So in DMPK, we have in vitro ADME and the in vivo PK platforms. And in the biology unit, we have 3 main disease areas that we work on: malaria, TB, and AMR. And we have screening platforms for all 3, as well as a cytotoxicity cross-screening and enzymology platforms to support all 3 disease areas. And then we also have a data storage and sample management unit. So the rest of the talk will be focusing on malaria drug discovery, or at least H3D's efforts at this. And as already indicated by earlier speakers, malaria is caused by the Plasmodium, parasites of the genus Plasmodium. It has a complex life cycle taking place in the human host and parts of the life cycle taking place in the mosquito vector. So the ideal drug should be able to target the different stages of the life cycle, so to offer protection, combat the relapsing form of the parasite, and then treat symptoms and block transmission. So a multi-stage activity drug. And then the speakers that came before also spoke about the resistance, the issue of resistance in the field. So the new drugs should have a novel mode of action to combat this resistance. So I'll be taking you through a brief history of MMV048, which is where our quest to find these kind of drugs that have multi-stage activity and new modes of action, how we came to work in the space. So researchers at H3D a few years ago screened a compound library, a biofocus compound library. So a library of 36,000 compounds. It was a phenotypic screen against the parasite, as well as mammalian cells to screen for cytotoxicity. This led to the initiation of the SFK40 series with an early lead identified as MMV0576. And then subsequent hit to lead and lead optimization identified MMV048 as a late lead, which went into preclinical development in 2012. So Brice briefly mentioned this, and then this was the first drug molecule to go into first-in-human trials on the African continent. And the other cohort was in Australia. The drug progressed to phase 2 clinical trials, which took place in Ethiopia until the study was shut down. So much as MMV048 never progressed further in the pipeline, it was considered a success for H3D because we were able to take a drug molecule from the bench into the clinic. And then another interesting thing about MMV048 is that it demonstrated multi-stage antiplasmodium activity. And subsequent target deconvolution studies with resistance selections and chemoproteomic analysis indicated Plasmodium kinase PI4K as the primary target. And in enzyme assays, we were able to show potency with an IC50 of 3 nanomolar. So MMV048 was shown to be to hit PI4K. PI4K is the primary target. So this then opened up an interest in targeting Plasmodium kinases for drug discovery, because currently there are no kinase inhibitors in clinical use for malaria. And as we saw, that MMV048 made it into the clinic. So this actually shows that it may be a good strategy targeting the kinases. So just to go briefly into why Plasmodium kinases are interesting targets, they are essential in multiple stages of the parasite, and kinase inhibitors are already in use for other disease indications, notable of which is the oncology field, and a lot of the inhibitors in clinical use are ATP competitive inhibitors. So the ATP binding site is highly druggable, and it is conserved across different species. So this presents opportunity for repurposing and repositioning starting points. But even given that as an advantage, it can also be viewed as a challenge of some sort, because minimizing off-target activities against the human kinases then becomes the challenge during the drug discovery process. But then there's significant phylogenetic diversity between the human and malarial kinomes, which present opportunity for selective targeting of the Plasmodium kinase. So just to demonstrate this, on the y-axis you have the different organisms, and with the human and Plasmodium parasite, Plasmodium falciparum included there. And then color-coded are the different kinase classes. And then you have the percentage kinome coverage at the bottom panel. And then what I would like to draw your attention to is in the, this is a pointer, right? So the tyrosine kinases that are the most targeted in the human kinome are absent in the Plasmodium falciparum. And you have the atypical Plasmodium kinases. So there's no clear orthologs between the human and the Plasmodium within these classes. And then you also have the fix that make a large proportion in the Plasmodium kinome. So this actually illustrates that there could be opportunity to selectively target the Plasmodium kinases for new drugs. And then moreover, quite a significant proportion of the Plasmodium kinome seems to be essential, at least in the asexual blood stage, which causes the clinical symptoms of malaria. So with this in mind, we've dedicated the last few years to identifying novel chemotypes and validating new kinases to target for the development of new antimalarials. So just in the search of chemical matter, a set of 84 human kinase inhibitors, which are either clinically validated or in preclinical development, was screened by a team at GSK. And so these compounds were screened against the asexual blood stage, and the identified antiplasmodium hits underwent a chemoproteomic study to identify potential kinase targets. And then another set is the KGCS set at the SGC. So this is a set of 187 human kinase inhibitors that have a narrow spectrum of activity. And then they were also screened against the Plasmodium parasite, and 25 phenotypic hits were identified. So the data from GSK was shared with H3D, and then together with the phenotypic hits identified from the KGCS set, we then performed additional profiling and in an effort to identify the mode of action for all these hits. And then in cases where a kinase is the primary target, then we develop an assay to incorporate into the drug discovery pipeline at H3D. So I will just take you through this type of work using sapanisertib case study. So this is some of our published work. So this compound was one of the kinohits that were identified in the GSK set. It is in the clinic for oncology indications, and it inhibits mammalian kinase, mTOR, which is a key regulator for a wide range of cellular processes, and then there's no clear ortholog for this kinase in Plasmodium. So it inhibits mTOR with a potent IC50 of 1 nanomolar, and then subsequent screening, we saw that sapanisertib inhibits the Plasmodium parasite with a nanomolar of 200, with a IC50 of 200 nanomolar in a 48-hour assay. And then chemoproteomic studies went on to show that the potential targets for this molecule is PI4K, a validated target, PI3K, and PKG. So PKG is another validated kinase target, and though there have been compounds that show potency against PI3K, we're not sure if it is actually a driver of efficacy in those chemical matter. So we did additional profiling, activity profiling, and then this compound does show anti-Plasmodium activity across all the life cycle stages, which is quite attractive. And then it also inhibits both PKG and PI4K. But when we do the conditional knockdowns in which you knock down the enzyme, and then that should increase sensitivity against the compound if the kinase that or the protein that you're knocking down is the primary target. So we see that there is an IC50 shift for PI4K, but not for PI3K and PKG. And I will not be going into the other studies that we've done, but basically what we were able to show with this compound is that it does have potent anti-Plasmodium activity, and PI4K is the primary target, but then there are secondary targets which include PKG and PI3K. So basically with this compound, we're able to probe a concept of polypharmacology where you try to hit two molecular targets with one compound to elicit a therapeutic effect. So one of the reasons why we're interested in polypharmacology as a concept is that it may have benefits such as reduction of resistance propensity and the potential to minimize pill burden and the costs associated with combination therapies. But obviously this is just the early studies of profiling. We still need to prove this. And then there are some challenges that come with this in that you have to reduce potency for the human kinase, which may really be a concern with mTOR because it has quite, with sapanisertib because it has quite potent mTOR inhibition. But off-target activity is not always a problem because, for instance, when you hit the human mTOR kinase, that may be a host-directed therapy for cerebral malaria, as this kinase is involved in modulating human host immune responses in this case. So we still need to do more studies to actually identify, understand the PK/PD drivers of efficacy for this compound and actually see if a dual inhibitor of kinases would actually be more beneficial than compound that hits a single target. Okay. So, but basically the point that we want to drive home is that there are opportunities for targeting Plasmodium kinases outweigh the liabilities. So they are highly druggable. And then because of the wide use of kinase inhibitors in the oncology field, you actually have multiple resources to use for drug repurposing and repositioning starting points. And then, as I indicated with sapanisertib, you have opportunity for designed polypharmacology, amongst others. So we are very cognizant of the liabilities that come with this, but we still think there is space and future for targeting Plasmodium kinases in the malaria drug discovery space. And then I'd just like to thank our collaborators and my supervisors who gave me an opportunity to come here. And yeah, thank you. I'll pause here for questions.
Thank you very much. Are there any questions? Please, oh, sorry. Well, can I start by, so this is not actually about the last part of your story. It was a hanging thread from the first part.
Okay.
But the MMV048, you said it had got to phase 2 trials in Ethiopia?
Yes.
What happened?
So the study was shut down because of COVID, but then there was also emerging safety issues with teratogenicity. So toxicity and
Okay,
fetal,
and that links really clearly to a theme earlier this afternoon about the problem about treating
Yes.
pregnant mothers. And
Yeah.
Okay. Okay, that's very interesting to hear. There any other questions? Please, do you mind using the microphone next to you? Thank you.
Okay.
Yes. Thank you very much. Thank you. Just curiosity, in case you didn't know me, I was one of the first students of Kelly Chibane when that 048 drug was discovered. So this seems like deja vu.
Nice.
Very biased about this one. But I'm just curious to know, so the study shut down, but you've got this potential drug. It has moved so far in the drug discovery journey. Any plans maybe of doing some form of modification, developing a drug analog or something that could maybe overcome whatever that has caused a stumbling block in that 2020 study, clinical studies?
So we do have a backup compound that we're working on right now. Yeah, I think so there is something happening. And then we're also looking at other chemical matter, as I mentioned. Still, I mean, we now have a fully fledged kinase platform where we're also doing, we're taking the target-based approach with the phenotypic approach to actually try and maximize the chances of a good output. But yes, we do have a backup drug that is quite advanced in the preclinical development as well. Yeah.
Thank you. Thank you. All the best.
Okay, thank you.
Thank you. Any other questions? In that case, thank you again.
Thank you.
So I'd like to finish by thanking all of the speakers for excellent talks and all of the audience for coming all this way to listen. It may seem strange to come to a cold northern country to hear about malaria, although actually Britain, from the time of the Roman occupation to the mid-19th century, Britain did have endemic malaria brought by the Romans, and it only disappeared because our changing agricultural practice eliminated the swamps in which the mosquitoes could grow. With climate change, it's probably going to be back. So I think we have a brief respite before we share your problems again, even domestically. So yeah, it's a global problem, and it's lovely to have a global gathering here tackling it. Thank you all very much, and I hope you enjoy the rest of the conference.